protective immunity in humans and what they can teach us for vaccine development. Arash?
Thank you, Rajan. I think after 2 years, one would get faster, but in sharing screens and stuff. But anyway, I'd like to thank Rajan, Chris Walker, John for their kind invitation to speak today. And I hope you can see my screen. I am certainly indebted to a lot of my good close friends and colleagues, not only with respect to tools, reagents, and collaborations that we have that I'd be able to give you this talk, but also through the recent endeavor with the U19, with Nagla Shukri in University of Montreal, Rama Amara here at Emory, and Nutan at Cairo University in Egypt. But I'll also bring in a lot of evidence from my great collaboration for years with Chris Walker and Rob Honegger and Amit Kapoor, and also Charlie and many others that I'll speak with you today. So we heard from Dave this morning that what is the evidence that vaccination can skew infection outcome toward resolution and away from persistence? Well, both as he mentioned as well, that the chimpanzees and human that have resolved their primary infection have a broad cross-genotypic protection against persistent and subsequent infection. Both the amplitude and duration of viremia is decreased significantly, and that has been shown with a number of colleagues, both in humans and chimpanzees. What's the evidence that T cells control the HCV infection? Well, we heard a number of from both Dave and Andrea Cox, and also in humans that there are virus-specific T cells are associated with control of primary and secondary infections, and Chris Walker's nice data with depletion of subsets such as CD4 and CD8 that resulted in prolonged or persistent infection. What about the fact that antibodies, whether they control HCV infection? Really great data again from Barbara Weirman, Andrea Cox, and others that have shown that you do see some antibodies and neutralizing antibodies, in those that go on to resolve. We, for the rest of the talk, I wanted to sort of focus more on the B cell side and T cell rather than the T cell, but we wanted to sort of address not just serum antibodies, but actual antigen-specific B cells as well to sort of figure out whether, how they contribute to this infection resolution. So obviously the amazing cohort that Nagla Shukri and Julie Bruneau had sort of painstakingly put together for past decade or so. These are subjects who are coming into the clinic. They're negative for HCV RNA and antibodies. And obviously they're in people who inject drugs and susceptible groups, so that then they're followed. And as soon as they become positive, then they are stratified between those who go on and develop persistent infection or those that are resolved spontaneously. So we took advantage of this cohort with Nagla, and here I'm showing you serum antibody results to multiple genotypes, genotype 1, 2, 3. And the thing that sort of struck me when we were looking at this data is that those who resolved are shown here in the top, and in red, they're the ones who develop persistent infection. The interesting thing for me is that the antibody responses in these individuals that go on to resolve is actually pretty poor. They don't really make that much of a robust antibody response, at least detectable in their serum. They actually, no response is seen to the non-structural proteins. The ones that go on develop chronic, obviously, is a kinetic sort of issue, but they all seem to cross-react with multiple genotype, even though their original genotype of infection may be 1 or 2 or 3. So the second thing was that the ability of the serum antibodies from these individuals who resolve to neutralize Hep C pseudoparticles, again, was pretty poor. Whereas the ones in the chronic infection, as you follow up those subjects, about a year and a half, they start to have some better neutralizing ability compared to many of those who go on to resolve their infection. So we took advantage of a sort of a tool, just like how people have designed for MHC tetramers to pull out antigen-specific T cells. We went ahead and cloned the ectodomain of the glycoprotein E2 and tetramerized it, so that we can go on to show that we can stain and isolate and purify antigen-specific B cells. And these are actually can be pan-genotype. You can isolate B cells from genotype 1, 2, or 3 with the same tetramer. That allowed us to sort of kinetically, again, look at antigen-specific B cells and when do they come up, or do they even come up? And here was the interesting thing, that in those that go on to resolve their infection, we were able to see these antigen-specific B cells come up at least few months earlier than those who go on to develop persistent infection. So even though the serum antibodies produced is very poor, but nevertheless, we can at least visualize and identify these antigen-specific B cells in the resolvers. And the surprising thing, again, is that when we go on and clone multiple antibody-heavy and light chains from both cohorts, and then assess whether they can neutralize the sort of the pseudoparticles and so forth, the surprising thing was that the ones in the resolvers, even though they came up much earlier, a few months earlier than those in the chronic subjects, they neutralized the virus very similarly. So, so that tells you that these antigen-specific B cells come up. They do have some ability to neutralize the virus compared to their counterparts that are about a year later. And this is telling you that even though they have much lower somatic hypermutation because they are coming up earlier, nevertheless, compared to chronic subjects, they're still able to resolve the virus, at least in vitro, equally. The other thing that we noticed that was different between those who resolve versus those who go chronic is this presence of these activated T follicular helper cells that not only they are present in their numbers, but they're also functional by producing IL-21 and CD40 ligand, interferon gamma, and many others that I won't go into details about this. But that gave us a sort of a big picture view that in subjects who spontaneously clear, at least we were, here's the virus level in red. We see this sort of an early expansion of T follicular helper cells that we think presumably they interact and help these B cells through IL-21 CD40 ligand to come up earlier and to produce the relevant antibodies that are able to resolve and help the virus to be abrogated. But importantly, nevertheless, they seem to just disappear very quickly and very rapidly from the periphery. The causes of that is really, at least we don't know at this time. In those who go on to develop chronic infection, we see this absence of these T follicular helper cells. And we think that these B cells that are coming up, they're sort of helpless B cells. They come up, and importantly, even though they start off with the same family of genes as you see in those who clear the infection, yet they are somewhat ignored. And what you then end up with is a lot of different family, VH169, for example, that come up much later. And while they're still able to at least show neutralization activity, by then the virus, I think, has escaped and is too late. So, so far, at least from my perspective, is that these role of B cells or antibodies that we see in these subjects is just, a correlation, and it's not very robust either. I mean, I'm very disappointed in the amount of serum neutralization ability of these when you compare it to, for example, with SARS-CoV-2 patients, that their serum antibodies certainly is capable of abrogating virus in much lower numbers. But, is there any evidence that these B cells or antibodies actually have a direct role during Hep C infection? And there, we took advantage with, great collaboration that we have with Rob Honegger and Chris Walker. We've been following a cohort of women that are chronically infected with hepatitis C virus. And interestingly, when they go through childbirth, these pregnant moms, after they give childbirth, so here's the third trimester viral load, and this is the 3 months postpartum. There's a cohort of these moms that there's no change in their viral load. Yet there is another cohort of moms that there is about a 4-log difference in their postpartum viral load. Now, Rob has beautifully shown that there is some T cell activation, T cell control that happens in these moms. Another important caveat that you should be aware is that through the GWAS studies with the IL-28B, there seemed to be a very strong correlation. The moms that there's a decrease in the viral load happen to be a CC haplotype, whereas the non-responders are either TT or a CT haplotype. But nevertheless, we took the serum from these controllers and non-responders and asked, well, is there anything in the serum, presumably antibodies, that would then show a difference in their ability to neutralize the virus? And indeed, in these elite controllers, in these moms, postpartum, you see a very strong increase in the breadth of the ability of the serum antibodies to neutralize multiple genotype of the viruses in vitro. So, do these serum antibodies then, do they apply any pressure in vivo to the HCV genome when after the delivery? And again, Rob painstakingly took and sequenced every one of these moms, and the changes, the non-synonymous changes that you see throughout the genome is shown in red, with the ones that are synonymous mutations in blue. But importantly, what you can see that majority of the non-synonymous mutations are occurring at the glycoprotein level, the E2. Whereas the synonymous mutations are just equally appearing throughout the structural and non-structural genome. What is also very interesting is that if you look at these mutations that are occurring, whether they're in a predicted MHC class 1 binding versus not, you can see that in the structural genes, these mutations that are correlating with a decrease in viral load, they happen to be in both class 1 binding epitopes and outside class 1 binding epitopes. These are presumably your antibody pressure and not CTL escape. Whereas in the structural genes, you can see, in the non-structural genes, you can see that the only thing that correlates is only mutations in the MHC class 1, the CTL escape, and not outside that region. So when you look at the sequence of these things a little bit more carefully in the glycoprotein, here with the elite controllers versus non-responders, there are two things that come out that are very sort of interesting and exciting. One is that you see a lot of yellow in the hypervariable region 1. These are amino acid substitutions that are within MHC class 1 epitope, and you see them in both elite controllers and non-responders. And actually the ratio of this is, you have 50 or so mutations in yellow to 10 mutations in green. So 5 to 1. But if you look at the front layer of the glycoprotein, that changes dramatically, and majority of these things are outside MHC class 1, presumably your antibody pressure occurring here. And in the back layer, these happen to be all yellow again, that are indicating that be a CTL escape. And you don't see any of these changes in moms who there's no sort of drop in their viral load postpartum. Now, if you put these mutations, great help from Joe Marcotigiano, who recently solved not just the structure of the glycoprotein E2, but in complex with the receptor CD81. And the picture looks like this. So this in tan, what I'm showing you is the front layer of the glycoprotein E2. And in dark gray is the back layer. So right off the bat, you see that this segregation of these green mutations, which are presumably occurring due to antibody pressure, they're all in the front layer. And the CTL escape mutations that are happening are in the back layer. I'd like you to focus on when we now put the receptor, the CD81, onto this complex. So this is what you see on a virion as the native form of the glycoprotein E2. But two things happens, and one is that this loop that you see here, which we refer to as a CD81 binding loop, could be a fusion peptide, but further evidence is needed. But nevertheless, you'll see that this thing is sort of in a closed, sort of a conformation. But as soon as it sees the receptor CD81, it pops up. And not only that, these green changes that you see, they all come together and coalesce where exactly CD81 is interacting with. And you can see that here. In blue is the CD81. You can see this loop sort of jumping up, and you see these sort of mutations that are really interacting closely with the CD81 binding. They come coalesce together and sort of, So this sort of, at least to me, in a very, naive way of looking at this structure is that, there's a lot of flexibility in the ability of this E2 glycoprotein to interact with CD81, and for these mutations to be sort of focusing on where the CD81 interaction takes place. Obviously, these mutations don't abrogate 100% interaction with CD81, because then there will be no virus. But nevertheless, they presumably have direct interaction and effect on the viral load. The second part of, sort of seeing whether there's a direct evidence for antibodies or B cells is to take advantage of the recent system that Amit Kapoor and Charlie Rice have shown using this rat Hepatitis as a model system to study this. Now, genome organization of this rat Hepatitis is identical to Hep C. And more importantly, Amit has shown that it develops a persistent chronic infection in rats, and Charlie's group has shown that they can be infected, mice can be infected, and again, a good thing that with Chris Walker's result in chimpanzees showing that any sort of a transient CD4 ablation causes the virus to become persistent in these mice as well. So then what this allows you to have is a lot of advantage with, with a vast number of genetic mutations and alterations to assess whether, again, the B cells are important or not. So we took advantage of three such models. One is called the MUMT. So these MUMT mice, they do have T cells, but they're devoid of any B cells. There's another one, a transgenic hen egg lysozyme. There is T and B cells, but your B cells only make antibodies to hen egg lysozyme and not to any other antigens. And finally, there's this AID-CRE mutation in which, while you have both T and B cells, your B cells cannot isotype switch. So basically, all the B cells remain to be IgM positive, and they do not become IgG. In black, you see wild-type mice that they resolve their infection roughly around 3 weeks post-infection. But in all these three models, you see that the virus persists. In MUMT mice, where they lack B cells, they seem to become chronic. held transgenic B cells, you can see that again they're prolonged, and in AI-DCRE, where you don't isotype switch to an IgG B cell, they go persistent infection again. So these models clearly tells us that the B cells are having a sort of a contributing aspect to them when it comes to this infection. So what's the best way to design a vaccine when we don't know even if it will work? For part of this endeavor, we took advantage of this sort of, can we learn anything from a natural vaccine-like immunity upon re-exposure of HCV resolvers? And here, with a cohort that, we have that they go through a primary infection, they resolve their primary infection, they're exposed again a second time, but with a much lower amplitude and duration of viremia. Compared to those who get reinfected and they become chronic, we felt that we could assess the immune responses between these two, and looking at the robust versus failed immune response, then we can sort of calculate and measure how we can look at the immune correlates of protection in order for us to develop several vaccine modalities. And as you know, Egypt, about 25% of the population is chronically infected due to schistosoma vaccinations back in the 60s. But now the government is active in resolving their infection using the direct-acting antivirals. But we think that in this group, they will have a similar phenotype as the ones that failed in their multiple infections. So this hopefully will give us a sort of a more complete picture in how we can move forward. So as an example, looking at the vaccinees versus reinfected resolvers, and this, we took advantage of looking at these subjects who came in and were re-exposed and resolved their secondary infection, and compared that to what Andrea Cox and Eleanor Barnes have shown in their subjects in which they were used with the prime and boost vaccination strategy. So this data with the transcriptome analysis allowed us to look at the following. I apologize that it's busy, but I think the take-home message is pretty simple. So here we're looking at the primary infection versus the vaccine prime, and a secondary infection versus the vaccine boost. And this basically looking at multiple different immune cell types. And we're looking at the different times, either early acute, late acute, and follow-up versus the primary and secondary vaccine boost. And what I like to sort of explain and show you is that here during the primary infection, not much of a T cell response is being sort of looking at based on the parameters shown here. But upon secondary infection, you get a relatively robust T cell response. In the vaccinees, you get a very robust T cell response. On the other hand, there's a complete, the glaring omission is the B cell or antibody responses that you see in the vaccinees compared to those that you see in the secondary infection. Is an example of 3 of such individuals that have been exposed and resolved. Some of them have pre-existing or memory T cell responses, and some of them, they're induced. These individuals have, in their antibodies, they can target glycoprotein E2 and non-structural proteins such as NS3. They have a very good neutralizing ability in their serum. And you see a sort of an induction of antigen-specific B cells that come up post-secondary infection. So how do you design? This is Rajan asked me, and it's actually a very tough question to ask, but how do you design and test the vaccine, candidates? And I guess in my sort of approach is to throw everything in the kitchen sink. As long as this kitchen sink only involves T cells and B cells, because the minute it involves NK cells, I think I'm going to retire and go fishing. But the reason we're going to try this approach is that with the help of Rama Amara here at Emory University, we can test the prime boost and coming in with a protein adjuvant, and to see whether, if we can hit this with a hammer, can we get a result that's encouraging? So the rationale for this is the following. In the SIV model, Rama has shown nicely that if you come in with DNA and MVA, sort of a boost, you see a log increase in your antigen-specific T cells. And these involve both CD4 and CD8 responses that are multi-functional based on various cytokines that they make. So there's a very robust T cell response that you can sort of induce by this approach. But importantly, if you now have your T cell responses at this stage, and now you come in and you provide a protein boost, you get about a 45-fold increase in your antibody responses as well. So then that helps us with the efficacy challenge model in the SIV, that you can see that in terms of the number of challenges that it takes to develop a productive infection, it becomes a very sort of a robust difference that you can see with this approach. So taking some of the lessons from the SIV and HIV models, we went ahead and used various immunogens, VLPs, virus-like particles, and the soluble E1, E2, and non-structural proteins, and sort of putting this together. At least the preliminary data is encouraging, which is expression of these forms, such as VLPs. Here you can look at with anti-E1 or E2 antibodies that you're producing them both in the cell lysates and in the supernatants. The secretory ones and E1, E2 also seem to be functional, and the non-structural protein expression, here you're looking at the NS5A, again shows that is working, at least at this stage. I'd like to end with this notion that at least this paper game that my daughter used to play with me to tell me which kind of ice cream I was able to have. I'm starting to think that the immune system, the plasticity that it provides, also it tries to give you the flavor. In some instances, depending on models, depending on subjects and prior infection and so forth, you may need one, or you may need 2 or 3 of these things to come in and help you with the resolution infection. So hopefully, with some of the efforts that we are undertaking, we can at least invoke some, or if not many of these things for a better outcome in the end. So with that, thank you very much, and happy to answer any questions.
Thank you very much, Arash. We have time just for one question, brief answer. Comes from Jasara Muhammad. For persons who have been treated with DAM medications, if later re-exposed or reinfected, is there any immunity or clearance observed?
Yeah, that's a great question. If at least the data suggests that if you resolve your infection taking these rather expensive drugs, and you get exposed again the next day, you will be reinfected, and so there's no protection immunity offered by taking these direct acting viral drugs. So the answer is no, you can get reinfected the next day.
Right, thank you, Arash. Yeah. So let's move on now to